This talk is particularly about gene expression and gene regulation. I'm offering a course in the Department of Biochemistry under this title. Uh, Already we have discussed that gene expression, whether it is prokaryotic or eukaryotic, is controlled at the transcription level, majorly at the transcription level. And in prokaryotic gene expression, there are certain operons are there, and then there is a regulon. Today we will especially discuss about major shifts in bacterial transcription with reference to gene expression. Uh, Bacteria control the transcription of a very limited number of genes at a time through the use of operons. More radical shifts in gene expression require some more fundamental changes in transcription machinery. And there are three of the major mechanisms. So these three major mechanisms, they include this sigma factor switching, RNA polymerase switching, and anti-termination. So we will discuss one by one all of these. The first one is the sigma factor switching. I will give some examples in this case, but first to review what sigma factor is. This factor is part of the RNA polymerase in prokaryotes. Prokaryotic RNA polymerase, when it comes to the promoter region, it needs sigma factor. Sigma factor actually is a loading factor of the RNA polymerase. It is not required for elongation of the transcription RNA polymerase, but it is required for loading of the RNA polymerase on the promoter in case of prokaryotic cells. So just by switching the sigma factor, the change in gene expression can take place. So lambda phages, there are different types of bacteriophages. These phage infection by bacterium, what it does is it subverts host transcription machinery. So these viruses, they don't have any life by themselves, but as soon as they come in contact with their host, they they overtake the machinery, and for example, the transcription, all the basic machinery of the transcription and translation they use from within the host cell. So we will see one of how this phage infection it controls the bacterial transcription or for its own synthesis of its own genes just by using the sigma factor. In process, it establishes a time-dependent or temporal program of transcription. So there are early phase genes that are transcribed, then there are some later genes, and then further late genes in the infectious cycles that there is no longer transcription of the host genes. Only the phage genes transcription goes on in case of phage infection in the late period of the phage infection. So these changes in genes that are transcribed is caused by change in the transcription machinery in RNA polymerase itself. So sigma is the key factor in determining specificity of T4 DNA transcription. T4 is one of the viral virus, and this T4 DNA transcription is also controlled by sigma. To shift the transcription process, sigma is a likely candidate. Similarly, SPO1 phage in Bacillus subtilis, this SPO1 has a large genome and it also has a temporal program of transcription. But this SPO1 is present in its host is Bacillus subtilis. So this figure you can see over here, the early genes that are transcribed by using the host RNA polymerase, and the blue one here is this one. This is the sigma factor, host sigma factor. In the early part, which is in the first 5 minutes, expression of early genes take place. After 5 to 10 minutes, expression of middle genes takes place. So in the middle gene, there is a protein, GP28 protein, that is synthesized. And now this GP28 is similar to the sigma factor. So this protein uh, rides over the RNA polymerase and transcribe the genes which from the SPO1 from the viral genome, and which genes are required for virus itself. And after 10 minutes to the end, late genes are expressed, and those late genes also contain, so there are two proteins which are synthesized in the middle when the middle genes are transcribed, and these two proteins, they're also called as GP, and these proteins, GP33 and 34, according to their molecular mass, so these two factors, they join together to just acting like a sigma factor, and thus this phage does not carry its own RNA polymerase, but it uses host RNA polymerases just by switching the sigma. So rather than the sigma, host sigma, it has made its own factors which act like sigma. And Bacillus subtilis RNA polymerase is hollow enzyme, is closely resembles to E. coli. It consists of beta, beta prime, two alpha subunits, and one very small omega polypeptide. Its primary sigma factor is 43 kDa, whereas in case of E. coli cell, the primary the sigma factor is 70 kDa. 70 kD. It's not 70,000 kD, but it is 70 kD. And actually, I think this D is redundant in all that. This K is redundant rather. So this is 43 kD, this is 70 kD. And this 43 kD is a smaller one, but it requires an additional factor which is 20 kD. And this 20 kD factor is, this subunit helps prevent the binding to the non-promoter regions, meaning this 20 kD factor along with 43 kD factor in Bacillus, it helps in the, it increases the specificity of the RNA polymerase to the so that it can bind to the certain specific places. So this figure shows subunit compositions of RNA polymerases in SPO1 phage infected Bacillus subtilis cells. So these polymerases, they were separated by chromatography and subjected to STS phage. So here there is an enzyme B and there is an enzyme C. Enzyme B is the first lane, this which lane it shows the core subunits beta prime, beta alpha and omega, as well as GP28. And enzyme C contains core subunits GP34, 33, and the last two lanes contain separated delta and sigma subunits respectively, and those subunits which are present in Bacillus subtilis. So this switching is directed by a set of phage encoded sigma factors that associate with host core RNA polymerase. These sigma factors change the host polymerase specificity, as I mentioned earlier in the figure, that GP28, 33, and 34, they switch the transcription of the viral genome for its own synthesis of its own genes, which are required in the middle and late part of the cycle. Sporulation, during infection, this SPO1 changes the specificity of the host RNA polymerase. Some type of mechanism applies to changes in gene expression during sporulation. Bacteria can exist indefinitely in vegetative state if nutrients are available, but on starvation conditions, this bacillus forms endospores. They are tough dormant bodies that can survive for years until favorable conditions return. So here A shows the vegetative cells dividing, and this B1 is the B is the sporulation with an endospore development at the left end. So during sporulation, a whole new set of genes are required and they are turned on. And many vegetative genes are turned off as they are not no longer needed. So this switch occurs largely at the level of transcription. Several new sigma factors displace the vegetative sigma factor from the polymerase core and direct the transcription of sporulation genes. Each sigma factor basically has its own preferred promoter sequence. Uh, More than one new sigma factors involved in sporulation. In fact, several of those they participate, such as sigma factor F, E, H, similarly sigma factor C, and this K, each play a role in addition to vegetative sigma factor A. And each sigma factor, it recognizes different set of promoters. For example, this vegetative sigma factor A, it recognizes promoters that are very similar to the promoters recognized by the major E. coli sigma factor, uh, with a -10 and -35 box consensus sequence. You already might know that this promoter in case of prokaryotes that contain -10 and -35 region. By contrast, these sporulation specific factors, they get recognized different sequences like sigma factor F. It appears first in the sporulation process in the 4 spore. So it activates transcription of 16 genes. These 16 genes are transcribed in this, including the genes that encode other sporulation specific sigma factors. And in particular, it activates SPO2R, which in turn activates the gene encoding sigma E in the mother cell. So together, sigma F and E, they put the 4 spore and the mother cell on an irreversible path to sporulation. Apart from this, so there are certain genes with multiple promoters, and some sporulation genes must be expressed during 2 or more phases of sporulation when different sigma factors predominate. So genes which are transcribed under different conditions are equipped with 2 different promoters. Each promoter is recognized by one of the different sigma factors, which ensures their expression no matter which factor is present. And it allows for differential control under different conditions. Here in the figure you can see overlapping promoters in Bacillus subtilis SPOVG. This P1, it denotes the upstream promoter recognized by sigma B. And this is the -10 and -35 region in the P1, which is recognized here, and the factor is the sigma B factor here. And P2 denotes the downstream promoter recognized by sigma E factor. So these two promoters, they overlap with each other. Now it depends just by change in the sigma factor, whether it is sigma B or sigma E, the RNA polymerase will bind to that particular uh, promoter and start its transcription. Hence, specific genes are transcribed. And this figure, it just shows the specificities of sigma B and E. This is an experiment where the researchers, they purified the S factors sigma B and E by electrophoresis and tested them with core polymerase using a runoff transcription assay that I already discussed in one of my earlier lectures. And this lane 1 containing sigma E, it caused initiation selectively at the downstream promoter as shown in this figure in P2, and similarly sigma B, it saturates with P1. So in this case you can see what is the difference between two and where they do transcription. This is another example of gene expression where if some bacteria, they are under heat stress. So in that case, the proteins which are expressed, they are called as molecular chaperones. What happens is that in heat shock, the proteins of bacteria, they are misfolded. And these chaperones bind to those partially unfolded proteins by heating and help them to fold properly again. So genes encoding these proteins that help cells survive heat are called as heat shock genes. How they are expressed? I give you the example of E. coli cells. Their normal growth is at 37 and high temperature, for example, 42 degrees centigrade. Normal transcription ceases or at least decreases at high temperature. And the synthesis of 17 new heat shock transcripts begin to counter this heat shock. And these transcripts encode molecular chaperones and proteases that help the cell survive the heat shock. This shift in transcription requires the product of RPOH gene, which encodes sigma factor with a molecular mass of 32 kD. Hence, this factor is called sigma 32, but it is also known as sigma H, where H stands for the heat shock. So 32 is the molecular mass and H for the heat shock. So you can say it as a sigma 32 or sigma H is the same, and it is the product of RPOH gene. So these 17 genes are expressed just by changing the sigma factor. So you don't have, the cell doesn't have to shift, use some repressors, activators and all that. They just change the expression just by having this sigma factor. Other sigma switches, heat shock response is controlled by 32, and this I already have discussed, and accumulation of sigma H with high temperature is due to stabilization of sigma H and enhanced translation of mRNA encoding sigma H, which is quite obvious. But responses to low nitrogen, starvation stress and other stresses, there are other sigma factors, like during nitrogen starvation, this sigma 54 or sigma N. It directs the transcription of genes that encode proteins responsible for nitrogen metabolism. In addition, although bacteria like E. coli, which are gram-negative, they do not sporulate, but they do become relatively resistant to stresses such as extreme pH and starvation. The genes that confer stress resistance are switched on in stationary phase, which is non-proliferating stage of the E. coli cells by RNA polymerases bearing sigma S or sigma 38. So all these examples are for the fundamental coping mechanism. Bacteria tend to deal with changes in their environment with global changes in transcription mediated by shifts in sigma factors. And those sigma factors are controlled by some other small proteins, and those are called as anti-sigma factors. These proteins do not compete with sigma factor for binding to a core polymerase, but they bind directly to the sigma and inhibits its function. One such example is a product of E. coli RSD gene, which is regulator of sigma D, that regulates the activity of major vegetative sigma 70, the product of RPOD gene. So this anti-sigma factor, as long as E. coli cells are growing rapidly, Most genes are transcribed by sigma 70 and no RSD product is made. However, when cells are under stress by loss of nutrients, high osmolarity, high temperature, or some other, at this point a new set of stress genes are activated by a new sigma factor which is called as sigma S, which accounts for about 1/3 of total amount of RNA polymerase in the cell. This means that 2/3 of the sigma present in the cell is still sigma 70. However, the expression of genes transcribed by sigma 70 has fallen by over 10-fold. So I just summarize it once again that when cells are growing rapidly, sigma 70 is the main promoter, is the main factor for transcription. Under stress conditions, sigma S is there, but still sigma 70 is still being expressed. But the expression of the genes is fallen by over 10-fold. So what is responsible for that? So different experiments show that it actually is the factor which is RSD, and this RSD factor binds to the sigma 70 and prevents its association with the core polymerase, thus acting as the anti-sigma factor. This anti-sigma factor is, maybe nature has made this because it is easier to control the expression at spot rather than switching it from a longer pathway. So if anti-sigma factor is synthesized and all of a sudden it can bind to the sigma factor, it can control that. Now this control it is possible that the cell doesn't need this control. So what it should have done, it should take out that sigma factor. So for that purpose, there are anti-sigma factors. Another example of this anti-sigma factor is in Bacillus subtilis. SPO2AB is an anti-sigma factor that binds to and inhibits the activities of the two sigma factors which are required for sporulation, like sigma F and sigma G. But another protein, SPO2AA, and that protein binds to the complexes of SPOAB plus sigma F and sigma G and releases the sigma factors, thus counteracting the effect of anti-sigma factor. And you might be surprised to see anti-sigma factor SPO2AB can also act as an anti-sigma factor by phosphorylating and inactivating SPO2AA. So it can do it directly by phosphorylating and it inactivates SPO2AA. Thus it is called anti-sigma factor. Next part is the RNA polymerase encoded in phage T7. T7 is another phage, a small genome and fewer genes than SPO1. And these phages have 3 phases of transcription, classes 1, 2, and 3. Of the 5 class 1 genes, gene 1 is necessary for class 2 and class 3 gene expression. How it goes is shown in this figure. Here there is a sigma factor and the RNA polymerase as usual for using the host RNA polymerase and sigma factor for class 1 genes. Then class 1 transcripts contain phage RNA polymerase. So in this case, it is not controlled by the sigma factor, but the phage makes its own RNA polymerase. And this RNA polymerase then make the further genes which are required in the late transcription for the class 2 and class 3 transcripts that are made with the help of this phage RNA polymerase. Now, example of Lambda phage, which is quite, you might have familiar with that, this temperate phage. I already discussed in earlier lectures its life cycle and all that. This Lambda phage has 2 paths of reproduction. One is the lytic mode, another is the lysogenic mode. And lytic mode, it acts as a virulent phage, and lysogenic mode, DNA is integrated into the host genome. This is the life cycle. So this at infection, its DNA goes inside the bacterial cell and it is circularized. So this red one is the phage DNA, and this virus is also very simple as all viruses. It has outer coat, it has head, tail, and its genome inside. So it is inside in the linear form, goes inside in the linear form and then circularizes. Here is the decision point. It may go in the lytic mode where this its genome is replicated and its head and tails are synthesized and new viruses are synthesized. So and this is lytic mode. Whereas in other case, it may be integrated into the host genome and go on and on with several cell divisions, several generations. And upon induction by some way, by UV light or something I will discuss shortly, it may be converted to the lytic mode. So what is controlling this lytic and lysogenic mode? Basically, it is a 27 kD phage protein which is called as lambda repressor or C1. It appears to bind to two phage operator regions. C1 actually it shuts down transcription of all genes except its own and some of the genes which are required for lysogeny. And when lysogeny is established, the phage DNA is integrated into the bacterial genome. The phage DNA in the lysogen replicates along the host DNA. And this lytic reproduction cycle of phage lambda has three phases: immediate early, delayed early, and late. Genes of all these phages are arranged sequentially on the phage DNA as shown here. As you can see, these are the lysogeny genes. These are the genes for DNA synthesis, late transcription, recombination, head genes, tail genes, and this is a cause site here, which these are cohesive ends on both sides, and this DNA when it goes inside the cell, it is circularized at the cause site, and these genes are placed on the its circular form is placed like this. So you can see that these are different colors they are shown. And these are the head genes and these are the tail genes. So important parts of here are this C1, which is responsible for this C1 is responsible for synthesis of, it is a basically lambda repressor, and then C2 and C3 helps this, and we will also see this CRO gene. CRO gene is also important in the control of this lysogeny and lytic mode, because this CRO is a repressor of C1. It means it is a repressor of the repressor. And then there are proteins which are required, and especially two important sites. One is the N and other one is the Q. These N and Q sites, they are called as anti-terminator sites, as we will see how they interact in the expression of genes in this case. So this anti-termination is a type of transcriptional switch. This is our third example of transcriptional switch. The host RNA polymerase transcribes the immediate early genes first. A gene product serves as an anti-terminator that permits RNA polymerase to ignore terminators at the end of immediate early genes. And same promoters are used for both immediate early and delayed early transcription. And late genes are transcribed by another anti-terminator. It will be more clear from this picture over here. One of the two immediate early gene is CRO. CRO codes for repressor of C1 gene, as I mentioned, and lytic cycle continues to move forward. There is N, as I mentioned in the last figure, that N is the anti-terminator region, and due to its anti-termination properties, all these genes are transcribed. And similarly for the Q gene here, Q site, the head and tail genes are transcribed in the late phase of the viral cycle. So anti-termination function, how it functions. Uh, you might have noticed that in case of promoters and operators, there are OL and PL, which are leftward promoters and operators, and OR and PR, which are rightward promoters and operators. Here, N anti-termination factor, which is present here, uh, this includes leftward promoter PL and operator OL and transcription terminator. Here the transcription is terminated. So what happens here is, if the product of this N is not there, this transcription is terminated at this place. Why it is terminated? As seen, you can see in this case it is terminated. Because there is a hairpin type of structure which is transcribed into the messenger RNA, which gives the message for the RNA polymerase to terminate, as in case of prokaryotes. So this is no longer transcribed further. But when this N is transcribed and it is there, so this N product binds over here, and when this N binds the transcript of N utilization site, which is called as NUT site, it interacts with the protein complex and changes its conformation, and this transcription signal here, that is no longer transcription termination signal is no longer functional, and transcription continuously goes on. And there are 5 different proteins that collaborate in anti-termination at lambda intermediate early terminator, NUSA, S10, N, NUSB, they bind to box A and box B, and N and NUSB bind to NSA and S10. I think it is quite confusing here, but it is more clear in this figure. You can see here, this is NSA and this is N, and this is the termination signal, and this is the box A part, and this is the box B part. So there are certain proteins, when those proteins are synthesized, those proteins bind at this place. So when these proteins are not there, this whole complex is not there, this is a weak non-processive complex, and RNA transcription is terminated. But once these proteins are placed in this box A in the messenger RNA, and they have interacted with the interaction with the RNA polymerase, along with the N and the NUT site, there is a strong processive complex, and transcription goes on. Similarly, for anti-termination for Q, you might have remembered that head and tail genes, there is a Q site in the late region, it is present. This binds to Q binding region in the QUT site, similar to NUT, as RNA polymerase is stalled just downstream of late promoter. So binding of Q to the polymerase appears to alternate the enzyme conformation so that it ignores the terminator and it transcribes the late gene. How this lysogeny is established? This phage establishes lysogeny by causing production of repressor to bind to early operators and preventing further early RNA synthesis. And delayed early gene products are used. Integration into the host genome takes place, and products of C2 and C3 allow the transcription of C1 gene. And this promoter, it is named as PRE, E for establishing the lysogeny. So here you can see this is RNA polymerase. It binds to the promoter PRE for required for establishing lysogeny, and these two small balls over here, these basically this is the C2, and this is the product of C2 in this direction. This C2 binds here, and this C2 allows the RNA polymerase to bind to this PRE. So PRE can, at the PRE, this RNA polymerase can bind with the help of this C2, and it allows the transcription of this C1 here. But it also regulates its own transcription, as you can see here, that if there is this C2 has a very high concentration, it may stop transcription by some method. So autoregulation of C1, it takes place. As lambda repressor appears, it binds as a dimer to the lambda operator, so which results in repressor, it turns off further early transcription and interrupts lytic cycle to turn off CRO, which is a very important, as product of CRO acts to counter repressor the activity, and it stimulates its own synthesis by activating PRM, M for the maintenance of the lysogeny. So lysogeny maintenance takes place with the help of these lambda repressor itself. Here, when lambda repressor it represses the synthesis of other genes, its own PRM is basically activated by this lambda repressor because of its specific binding. So this, if we fractionate this O operator, rightward operator, it is O1, O2 and O3. So when this is present only in O1 and O2 and O3 is clear, in that case this PRM is clear, and this RNA polymerase can bind at this place and start and continue transcription of the C1. But when this, if this OR3 is also taken up by the repressor, C1 repressor, this RNA polymerase cannot bind at this place. So this is, control is at this place. A dimer of two identical subunits, this repressor protein, this is just I already have explained, but there are two terminals, of course. One is amino terminal, is the binding DNA binding end of the molecule, and carboxy terminal is the site of repressor interaction that makes dimerization and cooperative binding possible. As you can see here, this is the DNA binding site, and this is the cooperative binding site one. And here there are two situations where this one is clear or it is not clear, as in this case there is no binding of repressor, and in this case there is a binding of repressor, as I mentioned earlier, how this repression of PR and PRM that takes place. This I already have discussed, that repressor binds to OR1 and OR2 cooperatively, but leaves OR3. So RNA polymerase to PRM, which overlaps OR3 in such a way that it contacts repressor bound to OR2. And this protein-protein interaction is required for promoter to work efficiently, and high levels of repressor can repress transcription from PRM. How this fate of lambda infection is determined, that whether it should go in the lysis mode or lytic mode or in lysogeny mode. So this balance of lysis and lysogeny is very delicate. Place phage particles on bacterial lawn, for example. If lytic infection occurs, progeny spread and infect other cells. If lytic infection occurs, progenies spread and infect other cells, and you can see circular holes in the lawn, which are called as plaques. So infection 100% lytic gives clear plaque, and plaques of lambda are usually turbid, meaning live lysogen is present. So some infected cells suffer the lytic cycle, others are lysogenized. So there is a battle between C1 and CRO. As I mentioned earlier, that C1 is the lambda repressor and CRO is the repressor of the C1. The C1 gene codes for a repressor, as I mentioned earlier, that it blocks these operators, so turning off early transcription. This leads to lysogeny, as in this case it is shown, and it is being inhibited. But its own C1 is being, its own transcription is going on. If C1 wins, it goes in the lysogenic mode, as mentioned in this case. The CRO gene codes for CRO that blocks this operator OR3 and OL3 during transcription. So this leads to lytic infection. And gene product in high concentration first determines the cell fate, whether C1 wins or the CRO wins. It depends on the concentration of CRO or the C1. And this lysogen induction, when DNA suffers with some damage, maybe UV or SOS response is induced, in that case one of the initial event is seen, and that involves the coprotease activity in REC-K protein, which along with other proteins also uh, degrades this lambda repressor. As you can see here, it is being degraded and lytic cycle is induced. So ultimately it depends on the environmental conditions, that what kind of conditions are there to whether it will go in the lytic mode or lysogenic mode. So if a cell is under stress, it will go in the lytic mode by turning off by degradation of the lambda repressor. With this, I conclude my lecture. Thank you very much.